Chapter 18 is all about the regulation of gene expression. Basically, how do we get particular protein products from our genes? How do we turn those protein products on and off in response to the environmental conditions as well as the conditions and needs for an individual cell? Both prokaryotes and eukaryotes can alter their gene expression depending upon what their environmental conditions are and how those conditions change. In certain environments, you need certain protein products, and in others, you can turn off that protein product and make others. It just depends on what's going on externally. With multicellular eukaryotes, gene expression is really important for the development of the organism and is also responsible for creating differences in cell types. So getting that signal that, hey, you're going to be a skin cell versus a liver cell and expressing that particular gene to make it a liver cell is controlled by gene expression and regulation. RNA also plays a role in regulating gene expression in eukaryotes, which we're going to find out a little bit later. This image is fruit fly larva that has been treated to show mRNA for 3 different genes, one that's shown by red, one that's shown by blue color, and one that's shown by green color. And by imaging these particular genes visually, we can look at different larvae between fruit flies, and we notice that larvae go through the same patterns of development as larvae to become fruit flies, and so it is very intricate in how those genes are expressed when they're turned on, and the patterns tend to be the same between each larva at similar stages in development. Let's talk about prokaryotes, and specifically let's talk about E. coli, which is a type of bacteria, as we work through our first examples of gene regulation. Natural selection has favored bacteria that can produce only the products that they need by the cell at that particular moment. It's not favorable to make every gene product that you can at one time. That would be really, really inefficient and unnecessary. So cells can regulate the production of enzymes by either feedback inhibition or by gene regulation, which we're going to go into. And if we're talking about gene expression in bacteria, that is controlled by the Operon model, which we're going to go through. Here's an example of regulation of a metabolic pathway. In this case, let's think about that E. coli that maybe is living in your colon. And depending upon your erratic eating habits, this E. coli may be well nourished, or it may go through periods where it needs to synthesize some of the amino acids it's not getting through your diet. So for example, let's say it's not getting tryptophan and it needs to make tryptophan in order to survive. So with the absence of tryptophan, we turn on the genes that create tryptophan. So we go through a series where you have tryptophan E, D, C, B, and A genes that are going to code for specific enzymes that take you from precursor all the way to your final product, which is tryptophan. Well, if that gene gets turned on, you're going to be making tryptophan and making tryptophan and making tryptophan. Eventually, you're going to have a surplus of tryptophan in that particular cell. Eventually, having a surplus actually inhibits the first enzyme in that step. So it stops that enzyme from being able to take your precursor into enzyme 2. Because it stops that step, you have stopped that entire pathway. You are no longer going to make tryptophan. So having an excess of the product in need can also inhibit making any more of that product. So it keeps it as an efficient machine. An operon is a cluster of genes whose protein product can be turned on or off depending upon what the organism needs. So think of it like a molecular on-off switch to get a certain product. There are 3 parts to any operon. They are the operator, which controls access of the RNA polymerase to the genes. So if that operator is functioning, it allows the RNA polymerase in so it can read that sequence of genes and make your product. If it's off, your RNA polymerase cannot get in. The promoter is where the RNA polymerase actually attaches to your sequence, and it's going to be upstream of the genes of interest. And then you actually have your genes of the operons. So this is that stretch of DNA that you need to read in order to make enzymes that are made by that particular operon. In the image below, we can see the trip operon is from here to here, and we see the operator, which is what controls whether the RNA polymerase can get in or not. The promoter tells that RNA polymerase, where do I bind, where do I start reading this? And finally, that stretch of DNA, which are the genes of the operon. These are going to code for whatever protein product you're in need of in your cell. All right, regulatory genes produce repressor proteins. So repressors are the actual off switch. And what these do is they interact with your operator. And when they're interacting with your operator, they will actually turn off the gene. So it blocks that RNA polymerase from transcribing that section of DNA to make your protein product. Repressors are typically on, but you can inhibit those. And if we have a co-repressor, we're talking about a molecule that actually coordinates and interacts with the repressor molecule, and it helps to switch the operon off. So our example down here, we see that our repressor right here is inactive, meaning it's not in the right shape to interact with our operator. When tryptophan interacts with the repressor, and it is a co-repressor, it changes the conformation, so it activates your repressor. In the active form, your repressor will travel to the operator and it will interact with the operator. When the repressor and the operator are connected, your polymerase cannot move through there. It blocks the RNA polymerase from moving forward and then reading those DNA or the genes of the protein or the enzyme of interest. So it does not allow the RNA polymerase to work, shuts down that protein product. As we kind of alluded to on the last slide, your trip operon is typically on, and the genes for tryptophan synthesis are transcribed. So you are making those enzymes that make tryptophan usually. It is typically in the on position. But if you have tryptophan present, it's going to bind to your repressor. So here we see the tryptophan binding to the repressor. When it binds, that activates your repressor, turns off the gene. Your repressor then interacts with your operator, turning it off, blocking the RNA polymerase. So what that basically does is it says, okay, we have tryptophan in abundance. There's no reason for you to synthesize more tryptophan. We already have enough. So we just talked about repressible operons. So that one is typically on unless your repressor binds to the operator and shuts off transcription. So your trip operon is a repressible operon, meaning it's on until the repressor binds with the co-repressor tryptophan and turns off that gene. The opposite of that is an inducible operon. This one is usually off, and a molecule called an inducer inactivates the repressor, turning on transcription. So think of the repressible operon and the inducible operon as being opposites of each other, whereas to turn off the repressible operon, you need a repressor. To turn on an inducible operon, you need an inducer. The lac operon is an example of an inducible operon, and the genes that it contains are ones that create enzymes that hydrolyze lactose. So it breaks apart the milk sugar lactose into its monosaccharides. So it basically makes lactase. By itself, your lac repressor is typically active, meaning your operator is turned off, it's bound to the operator, polymerase cannot come through and make enzymes to break down lactose. And the reason for this is you don't need enzymes to break down lactose if lactose isn't present. So if you have lactose present, that is an inducer that inactivates your repressor molecule, turns on transcription, you make the enzyme to break apart lactose. And this will be a little bit easier when we see a picture and we talk through that as well. And here it is. Okay, so this is a lac operon. So this is typically on, meaning your repressor is on, you are not making any of those gene products. The reason it's typically on and the gene is turned off is because you're not always taking in or consuming lactose. You don't need to make an enzyme for something that you don't have present all the time in your body. So this is an active repressor. If you get allolactose, which means you have lactose present, that allolactose binds with your repressor and it changes the shape. This shape can no longer interact with your operator, meaning now we've unbound from the operator. Polymerase can move right through there. It turns the gene on, it turns the repressor off. So now that lactose is present, I'm going to start making the gene product lactase. I'm going to start making that enzyme that breaks down lactose until I no longer have lactose present and it unbinds, and then your repressor goes back in and binds with the operator again, turning off the gene. Inducible enzymes usually function in catabolic pathways. So this is when you want to break something down. So think about having lactose present. If you have lactose present, you want to make an enzyme that breaks that apart into its monosaccharide. It breaks it down into a smaller molecule. Typically, if you want to make the enzymes on that gene or make that product, you need a chemical signal, such as the presence or absence of lactose. Repressible enzymes usually function in anabolic pathways whenever you're trying to build a molecule, such as trying to make tryptophan because it is not present in the body. The synthesis is repressed by the end product. So when you get a bunch of tryptophan, that actually binds with the repressor and causes the gene to shut off. Regulation of TRP and LAC operons involves negative control of genes because your operons are switched off by the active form of the repressor. Aside from negative gene regulation, we also have positive gene regulation. So some operons are controlled through a stimulatory protein, such as a catabolite activator protein or a CAP. What this does is it activates transcription. So it takes the DNA and it starts processing it into an mRNA, which can then be used to make protein products in the ribosome. A good example of this is E. coli. E. coli can have both lactose and glucose present, and it can function on both. But if both are present, it prefers to use glucose as its energy source. So if you have low glucose in E. coli, your CAP is activated. Cyclic AMP is the signal molecule that activates that catabolite activator protein. And it says, okay, glucose levels are low. We're going to need to synthesize some lactase so that we can start to break down the lactose that is present. We don't have enough glucose to survive. We need to use a different energy source. So your activated CAP attaches to the promoter of the LAC operon, and it increases the attraction for RNA polymerase, which means you're going to get faster transcription because of this. So RNA polymerase comes and adds RNA nucleotides, complementary RNA nucleotides to your genes of interest, which makes your mRNA strand. However, if you have high glucose levels, the E. coli would prefer to use glucose as an energy source, and when there are high levels, your catabolite activator protein, your CAP, detaches from the LAC operon, and then transcription for lactase, or the enzymes that break down lactose, decreases. So it says, okay, we have glucose present, we do not want to produce this enzyme that will break down lactose as an energy source. All right, so here we have an example of E. coli where you have lactose present and glucose is scarce. Because lactose is present, your repressor is bound to the lactose. It is now inactive. The repressor is inactive, which means we can make our protein product because it's not binding with the operator. So there is no glucose, so we have to say, okay, hurry up, speed up the process. We have lactose, we need an energy source, we need the enzymes associated with the lactose pathway. Cyclic AMP binds to an inactive form of your catabolite activator protein, which activates it. It then binds with the promoter region of your operon, and that says, okay, RNA polymerase, come over. We need you to really, really quick make an mRNA, complementary mRNA from our genes of interest so that we can synthesize the lactose enzyme pathway. If you have lactose present and glucose present, E. coli prefers to use glucose as an energy source. So your cyclic AMP levels are very low because we don't want to make the enzymes that break down lactose. We'd much prefer to break down glucose. So you have an inactive catabolite activator protein. So it's not causing transcription to be faster than in the example where glucose is scarce because we want to break down the glucose. However, you still have lactose present. So your inactivator is inactive, your repressor is inactive, excuse me. It cannot bind to the operator. So you're still going to produce enzymes to break down lactose because it's present, but we're not speeding that up because we would prefer to use glucose as our energy source. So organisms have an encyclopedia full of genes and possible protein products that they can make. So regulation has to be closely monitored for which genes are going to be expressed at which time. Some are only supposed to be expressed during embryonic development, some should be expressed later in life. And these differences between your cell types, so between liver cells and skin cells and brain cells, result from differential gene expression. So that's expressing different genes by cells with the same genome. So taking that gigantic piece of DNA and picking out the genes that code for what cell type you're going to be and what expression you are going to have. If you have errors in gene expression, this leads to diseases such as cancer, and the more we learn, the more we find that gene expression can be regulated at a bunch of different levels. Here's all the different places that we can regulate gene expression. So the first is chromatin modification. We can actually regulate, turn on and off certain sections of DNA by chromatin modification. We can regulate RNA and mRNA during transcription, RNA processing, and the transport to cytoplasm. We can also regulate RNA and hence the amino acids during translation, the final protein processing, as well as through transport to cellular destinations and whether we degrade that. There's a lot of different levels of where genes can be regulated and gene expression can be controlled. If your gene has a highly packed heterochromatin, it's usually not expressed. And the reason for that is it's packed so tightly that all of those enzymes you would need for transcription, such as RNA polymerase and primase, those cannot actually get into that tightly packed DNA. So transcription does not occur in those, or it doesn't happen as often. We can chemically modify the histones, and that is the DNA with the protein wrapped around the protein of chromatin. We can influence both chromatin structure as well as gene expression. And there's 3 different ways. Histone acetylation is the first, through methylation, and through the addition of phosphate groups, which is also called phosphorylation. The first method is through histone acetylation, and this is where you take an acetyl group and you attach it to the positively charged lysine in the histone tail. So this little guy would be considered a histone tail, and down here you can see we've stuck a little acetyl group on it. And what that does is it prevents those histones from packing super close together. So it loosens your chromatin structure, and because they're a little bit more separate, your enzymes for transcription can get in there. So your RNA polymerase can come in, your primase can come in. Those enzymes can get in, and you can actually get transcription of the DNA. It's not as tightly packed. DNA methylation does the opposite of acetylation. It condenses the chromatin. It causes tighter packing of the chromatin, which inhibits transcription. So you're not going to express the genes that are on this piece of DNA that's been methylated. We talked about genomic imprinting a while back, and this is how it actually functions. Your methylation will regulate the expression of either your maternal allele or your paternal allele of certain genes at the start of development. So basically turns off your maternal allele for a trait or your paternal allele, and then every cell that it divides into after that also has that same genomic imprint, meaning you're not going to transcribe both of those alleles from mom and dad at the same time. If we were to add a phosphate group, which is phosphorylation, this also loosens the chromatin, similar to how the acetylation works, which promotes transcription. So we can read those sections of DNA and produce a transcription product, an mRNA, from that. In essence, how those 3 mechanisms work is they are modifying your chromatin. They provide the initial control of gene expression by making a region of DNA either more accessible to transcription machinery or less accessible, meaning it's going to promote transcription and gene products to be made, or it will prevent transcription and gene products being made from it. Besides using transcription factors and polymerases to get at that gene of interest in the protein product, your eukaryotic genes are also have control elements, which are segments of non-coding DNA, which help to regulate transcription by binding certain proteins. So here we can see upstream of your gene of interest are enhancer control elements, and these are represented by gold. So these help transcription factors to bind, as well as your proximal control elements. And then we also, they say, okay, come in, bind, do your transcription, make your protein product, which we can see through our primary mRNA transcript, as well as our already processed section of mRNA. It has a poly A tail and the 5' cap. You also have termination regions, which are downstream, so past your gene of interest, that say, okay, stop transcribing. We're at the very end, and they help the RNA polymerase to cleave or to terminate transcription. As I showed in that past image, to get transcription started, RNA polymerase requires proteins called transcription factors to come in and help transcription be initiated. Your proximal control elements are located very close to your promoter, whereas your distal control elements or your enhancers are far away from the gene. They're usually located in an intron, which is considered your intervening sequence. It is not expressed. It would be removed during your RNA processing. So this is an example of a transcription initiation complex, and you have these activators that bind to your enhancers, and that stimulates transcription. And basically what happens is these activators bring your transcription factors in close to where your promoter region is, so it brings it in close proximity so that you can draw in that RNA polymerase and get things started. The bound activators cause mediator proteins to interact with proteins at the promoter, which again pulls in your RNA polymerase and says, hey, we're going to start transcribing this section of DNA. So they all work in conjunction with one another, but it is the enhancer that helps that piece of DNA to fold over so that you can bring all of these, the transcription initiation complex, in close proximity to the promoter. Transcription and chromatin condensing is not the only way that we can control what genes are expressed. We have regulatory mechanisms at various stages after transcription as well, or they're considered post-transcriptional regulation. And these mechanisms allow a fine-tuning of your gene expression. And this happens quickly enough that you can respond to environmental changes. In alternative RNA splicing, you can take the same piece of DNA and you can change which segments of that RNA are considered exons and which are considered introns. So by changing that, you change what your gene expression is and what your mRNA sequence is going to be, which will eventually, through translation, change what your protein product is. So here's our primary RNA transcript. We have genes 1, 2, 3, 4, and 5. Here I can see that 4 was read as an intervening sequence and was removed, and only 1, 2, 3, and 5 were considered exons or expressed regions. In this example, I see that 3 was read as an intervening sequence, so it was removed, and 1, 2, 4, and 5 were considered exons or expressed regions. So now we have different bases in our mRNA, which are going to code for different protein products. So we can read that same gene multiple ways. So the initiation of translation for your selected mRNAs can be blocked by regulatory proteins that bind to sequences or structures of your mRNA. So you can block it before translation even occurs, so translation will not occur. Also, translation of all mRNAs in a cell can be regulated simultaneously through different mechanisms. If translation were to occur and we make a protein product, we can also control whether that protein product remains in a cell. So if we don't need an enzyme anymore, we probably need to get rid of it so it doesn't continue working its magic. And to do this, we need a proteasome. So what happens is this little orange trash can looking guy is your proteasome, which is a giant protein complex. It's going to bind molecules and degrade them. If you have a protein that is no longer needed, you don't need this product anymore, you don't need this enzyme to be functioning, it gets tagged with multiple molecules of ubiquitin. Ubiquitin, once it's tagged, acts like a magnet to the proteasome. So the proteasome sees that ubiquitin and it says, okay, that one's been tagged for destruction. I'm going to chop it up into a million pieces, rendering it non-functional. So once those have gone through the proteasome, you're going to get protein fragments, which are just peptides at this point, and they can be recycled in the cell or used at another point. Back in the day, we noticed that not all segments of DNA were actually transcribed into RNA, and we were like, wow, this must just be a bunch of leftover DNA, part of our evolutionary history. It's just junk DNA. And now we're finding that those sections that we thought were junk DNA, coding for hence junk RNA or non-coding regions of RNA, actually plays a really important role in controlling gene expression. So we know that only a small fraction of DNA actually codes for proteins, rRNA and tRNA, and a significant amount of the genome may be transcribed into non-coding RNAs. We know now that those non-coding RNAs can regulate gene expression at 2 points, both mRNA translation, so when you're making your proteins in your ribosome, and also chromatin configuration. MicroRNAs are small single-stranded RNA molecules that bind to the messenger RNA, the mRNA. And their function is to degrade mRNA or to block its translation. And so what we're seeing here is you have this section of mRNA and it folds back in on itself, forming these hairpin structures. And once that hairpin structure has formed, you have a dicer that comes in, chops off that loopy end, which frees up the microRNA that we need. And this guy is looking for mRNA floating around or in the ribosome. And once it finds that, if there are more than 6 nucleotides in common, complementary, it can actually chop up that piece of mRNA, rendering it non-functional so it can no longer be translated into a protein product. Or if there is less than 6 bases, it will just bind to it. When it's bound, it cannot enter the ribosome and go through translation. It's blocked. So it's one way to control the expression of that mRNA transcript. Another mechanism of gene expression and the inhibition of gene expression by RNA molecules is called RNA interference, and that is caused by small interfering RNAs, and they play a role in heterochromatin formation. They can block large regions of your chromosome. Basically, they're turning off any RNA that is similar in structure to the RNA that they have. So these can block transcription from the get-go of specific genes. We're not talking about blocking at the translation level, but we're blocking those genes from even being transcribed. So we talked about gene expression and controlling which products are made within a cell, why that's important and where that happens. Now we're going to talk about how differential gene expression can lead to different cell types being formed in a multicellular organism. So we know that during embryonic development, your fertilized egg can give rise to a bunch of different cell types. You get your skin cells, your liver cells, your brain cells, and those cell types are then organized into tissues. Tissues are organized into organs, organ systems, and eventually the entire organism. Gene expression is what orchestrates this development, and it says which genes are going to be turned on at which time in embryonic development. So it's this really coordinated, concerted effort to make sure that the organism has the correct body plan. In order to get from a zygote to an adult of any organism, you're going to have cell division. So you're going to need a series of mitotic divisions just to increase the number of cells so that you have more cells and you add to those cells. You're also going to need cells to differentiate, and this is how we get cells that are specialized in both structure and function. Your muscle cells have a very different structure and a very different function than your skin cells do. And lastly, morphogenesis, and this is the physical process that gives an organism its shape. There are 4 things that control differentiation and morphogenesis, and that is cytoplasmic determinants, cell-to-cell signaling, determination, as well as pattern formation. We're going to go into each of those. Cytoplasmic determinants come from the egg cells, so they are maternal, and they can influence early development. They're distributed unevenly in cells, and because of that unequal distribution, you wind up with different effects. So in this image, we can see an example of cytoplasmic determinants. We have the yellow on one side and the green on the other side. And once fertilization occurs, we get the signal to go ahead and go through mitosis. So the nucleus divides, and when that cleavage furrow forms, notice that one side has more of those yellow dots and the other side has more green triangles. Well, they have a different distribution of cytoplasmic determinants, and that's going to be important when we start to, those cells start to develop and differentiate into different cells. They're going to get different signaling. The second is induction, which we'll talk about on the next slide, but you get a signal from one cell and it's passed to a neighboring cell, and they can actually alter through the signaling, they can alter each other's gene expression, which is called induction. So as we just mentioned on the previous one, the process of induction is where you have signal molecules from an embryonic cell causing transcriptional changes in nearby target cells. Remember, transcription is what gets us our gene products. We're going to get different protein products, which is going to lead to differentiation of the cells as well as specialized cell types. Determination is what commits your cell to its final fate. So once a cell goes through determination, there is nothing that will change what cell type it becomes. There have been studies where they take a cell that's gone through determination and placed it next to other types of cells, and they could not induce a change. They could not get that cell to change into the cells that are around it. Its fate was determined. It was done. This happens after that cell-to-cell signaling, which we see during cytoplasmic determination. And it is irreversible. If the cell gets a signal to become a skin cell, it will forever be a skin cell. And the cell differentiation is marked by the production of tissue-specific proteins, so what makes it unique as a cell type. An example of determination is in myoblasts, and myoblasts produce muscle-specific proteins and form skeletal muscle cells. So when a cell gets a signal from another nearby cell that, hey, you are to become a skeletal cell, it starts to produce this master regulatory gene, or MyoD, and this produces more proteins that commit that cell to becoming a skeletal cell. So these protein products are made. The MyoD protein is a transcription factor. It binds to enhancers and it causes transcription to happen. So it allows for DNA to be read and transcription products to be made. So once that determination step has happened, those protein products are made, the cell will forever be a skeletal muscle cell and it cannot become anything else. Pattern formation is how we develop our spatial organization of tissues and organs: up, down, left, right, supine, whatever. In animals, that begins with the establishment of the major axes, and we're going to see an example of that in the next slide. Positional information are molecular cues that control that pattern formation. So it says, okay, here's where you are located, little cell, compared to the rest of the body, compared to the body axes, as well as compared to other cells. So it's kind of like a you are here, this is where you're at, this is your position. And we have these substances called morphogens, and having a lot of morphogens in one area, not so many in the other, is how you can establish the embryo's axes. So figuring out which way is anterior versus posterior, as well as where other features such as legs and arms belong. So we've studied Drosophila a lot when it comes to pattern formation and morphogenesis, and we know that there are cytoplasmic determinants in the unfertilized egg that are going to determine the axes before fertilization even occurs. So here we see your egg cell is developing and going through the nurse cell. Eventually that follicle cell secretes that egg cell, it gets fertilized, and you get embryonic development. And notice the colors. So the head is represented by green. So because these cytoplasmic determinants, we know the head is going to be on one end, the tail on the other. We see the abdomen develops in orange, and that thorax, that middle section, is in brown. And that's due to those cytoplasmic determinants that were in the egg before fertilization even occurred. Some fun studies on fruit flies that we've done involve the bicoid gene. And the bicoid gene is what is going to determine where the head, or the anterior of the organism is. So if you have an embryo whose mother has a mutant bicoid gene, meaning it's not functioning properly, it's not going to have the front half of its body, and it's in fact going to have a tail on either end because it doesn't recognize where the head is. It can't establish that axis. And so there was an experiment done. We're visualizing the mRNA that codes for that bicoid gene. It says, hey, this is where the head goes. The anterior end does not have that, the posterior end does not have that. So here we see those cytoplasmic determinants, that mRNA that codes for here's where the head goes. And in a mutant who lacks this gene, we wind up with this result. We have an embryo that develops two tails, no head.